Earlier, Stefan Lorkowski, and I'm Professor for Nutritional Biochemistry and Physiology and the Director of the Institute of Nutritional Sciences at the Friedrich Schiller University in Jena. I am also the coordinator of the Competence Cluster for Nutrition and Cardiovascular Health, NutriCard, which is a joint initiative of the universities in Halle, Jena, and Leipzig. Our Competence Cluster NutriCard is a national corner pillar for nutritional research and is one of only 4 national centers of excellence in nutrition research and is funded by the German Ministry of Education and Science. The focus of our work is to better understand the complex interactions between our diet and the development of cardiometabolic diseases. And as you all know, more and more attention is paid on the microbiome and our diet, as well as host microbiome interactions, as there is increasing evidence that these factors contribute jointly and significantly to the risk of the so-called non-communicable diseases, such as cardiovascular diseases. And in fact, the metabolome is the functional readout of the information encoded in our genome, and metabolomics studies help us to better understand the complexity of the metabolism in our body, and more importantly, metabolomic studies are a vital and powerful tool of systems biology that allows to unravel the manifold interactions between our diet, our microbiome, our body, and the development of metabolic diseases. Therefore, in this webinar, the following speakers will provide deeper insights into the metabolomic studies in nutrition and microbiome research. I'm now very happy to announce Professor Dirk Haller from the Technical University Munich, Professor Dr. Lars Ove Dragstedt from the University of Copenhagen, and Professor Dr. Martin von Bergen from the Helmholtz Center for Environmental Research in Leipzig. The first talk is now given by Professor Dirk Haller on the topic Microbiome Signatures in Human Populations, a functional approximation to disease susceptibility. After each talk, we will have about 5 minutes for questions and answers, so please write your questions into the chat box. The question and answer sessions will be moderated by my dear colleagues, Dr. Sebastian Gottfried and Dr. Bijan Chatterjee. All presentations will be in English, and the webinar will close at 12 a.m. So the webinar will be recorded and will be available later via a download link. So please contact Biocritus for further information on this issue. Thank you very much for your attention, and now enjoy the webinar. And the first presentation by Professor Dirk Haller from Munich is given by an expert in nutrition and immunology. And I would particularly to point out that Dirk Haller is the initiator and spokesperson of the DFG-funded Collaborative Research Center on Microbiome Signatures. So please enjoy the first talk.
I would guess that everybody hears me now. Welcome to this. Pardon?
Great, Dirk. Dirk, it's great. It works.
Okay, very good. So welcome to this webinar. This is a new experience, and in light of corona infections, maybe we do this more often. So it's a pleasure to join this this exercise. And what I try to do in the next 20 minutes is I try to educate you a little bit on strategies to understand the function of the microbiome and effects on health. But what I really want to do is to give you also a little critical reflection on current concepts. So starting with a slightly historical perspective on the microbiome involvement, and we start usually with terminology. Microbiome as a term was, I would say, best coined by Joshua Lederberg and Alexa McRae already 2001, and they published this in The Scientist. Nevertheless, you have to understand that this term microbiome is already used since 50 years. The term microbiome is basically a shared term of microbiota, which describes all microorganisms in a given habitat. And the metagenome then includes their collective genomes in the totality. So if you combine these terminologies, you end up at the microbiome. But important to understand is that these two, I think very early on already pointed towards the importance of health and disease. And as you can see here, publication records in PubMed they exponentially rise, and we basically at the moment double every year the number of publications related to that, and everything is driven by technology. And when 2006 high throughput next generation sequencing technologies were available, the whole community started actually to describe in large details associations between changes in microbial ecosystems and certain conditions. Again, when you look at the publications and how you can stratify them, a lot is published on the gut, and that makes a lot of sense because the gut actually harbors the most dense microbial ecosystem, and more than half of the publications are related to the gut microbiome, and accordingly the gut is a central organ for orchestrating immune function, so the immune system is very much involved. Liver, brain, skin are also topics rising. And if we go a little bit towards here, the disease side, it's actually a lot related to inflammation, tumorigenesis, but also metabolic related diseases, obesity, type 2 diabetes, cardiometabolic diseases, and certainly the nutrition is impacting. So here you have a list of different diseases, and if you would look at the moment in PubMed, I think there is almost no disease or non-communicable disease which is at the moment not related to changes in the microbial ecosystem. I think what we all need to understand is that this ecosystem in the intestine is not a homogeneous ecosystem, and if you look at the distribution, spatial distribution between small and large intestine, there's already very clear that the density in the large intestine of bacteria, it's much higher than in the small intestine. Transit times in the small intestine is 4 to 6 hours after meal intake over a large length in the small intestine. That is certainly because we need to absorb nutrients. Then you have a large density in the large intestine, a large and extensive transit time, depending on what you eat as well, between 30 and 70 hours. There is basically retention time in the large intestine over a small location. At the end of the day, we defecate 70 to 500 grams, depending in average where you live. Scottish individuals, for example, are at the lower end with 70 grams per day. If you go to Central Africa, for example, we have 500 grams stool biomass every day. And you can think about where you are located in this scale. It's important, I think, to understand that the large intestine is a very unknown environment. And this is very simply already basically described by understanding what the composition in this large intestine is. And what you see here is an MRI detection of water, and water distribution in the colon environment is very inhomogeneous, and there is probably water pockets around, and many of reactions require a certain water environment. So again, having said that, we don't really understand the details of this environment, and this is very important when we think about bacterial metabolism, for example, and communication with the host. And what you see here is an analysis where bacteria are located, and you can see this is the epithelial surface in the large intestine. Green is mucus production, and you see that there is a thin layer of mucus which clearly separates bacteria from the host, and these bacteria are located in the outer layer of the mucus. So there is intense interaction. Nevertheless, there is also a clear compartmentalization of bacteria with respect to the host. So chapter 1, diversity of individuals, and that I think is something which comes up very clearly. And what I want you to show here is an example of a published Chinese cohort, and I compare this to a cohort which we are in the process to analyze. This is published in bioarchives and currently under review, what you should see here, this is 7,000 people, this is 2,500 people. And if you look at the bacterial classification along the taxonomy, you can classify them at the phylum level, which is a very broad classification, which at the end goes down to species and strain level. At the phylum level, the conclusion here is that the Chinese population is very similar to the population, for example, here in Munich, because the proportions of major phyla, Bacteroidetes to firmicutes, in both cases, firmicutes here, and the proportional variation within the population is very similar between Chinese and Europeans. And that's one take-home message. Nevertheless, there is also substantial, and I go back once, there's substantial variation at the species level. And this is at the moment subject of evaluation, trying to understand why at lower taxonomic levels we are so different. And again, this is the Chinese evaluation, this is ours. And at the end of the day, we and the Chinese more or less can explain between 9 and at max 20% of the variation within the populations. And you can already see here districts in the Chinese population study that the larger the location is apart from each other, the more it's going to impact microbial analysis, meaning that it's very hard to compare studies maybe in Germany, England, and China. There's other factors, lifestyle, physiology, disease, medication, which affects, in our cohort in Munich, is geographically very confined around area of Augsburg, Munich. And I want to talk a little bit of another factor, which is time of diffication. And what you see here is, for example, that circadian rhythmicity is something which is actually very much present in the microbial environment. Here described richness, the number of molecular species, that's diversity, and that's the two major phyla here. Green is a healthy population, red is a diabetic population. And you see this green in a normal population that you have a 24-hour oscillation very clearly in richness. And if you look at the major phyla, Bacteroidetes and firmicutes, they basically oscillate related to each other. If you have low levels of Bacteroidetes, you have high levels of firmicutes, and that changes over day. If you look at pre-diabetes or diabetes, especially diabetes here in red, you see that this harmonic oscillation is basically completely disrupted. So at the end of the day, this means that sampling time of the day is one aspect very important. Coming back to diversity of individuals, you see here the number of molecular species in different individuals. This is the richness depicted here. And you see that you have a distribution, everything between 100 species up to 600. An average is basically round about 380 species in one individuals. And we're looking here at almost 2,000 people. Having said that, diversity massively increases if you look at the strain level. And here are 3 species shown which represent parts of the microbial diversity at the species level. And you see 2 things. First, if you, for example, look here at Prevotella copri as an example, you see the phylogenetic tree, and we find many, many different bacterial strains here in the different populations. And there is quite a difference between strains, for example, in China, red, compared to other countries. So at the moment, we conclude that single strains dominate individuals, and that we have, again, massive subject and regional variations at this strain level diversity. And here is dynamics shown a couple of, or last year published from a group in Boston, in a population of diseased people, chronic inflammation. And if you look at the Prevotella copri here, this is normal individuals, this is individuals with ulcerative colitis or Crohn's disease, and you see that the individuals have quite massive and different fluctuations of one species over a certain period of time. So the microbial ecosystem is dynamic, and many factors contribute to variations. So when we do studies, I think it makes a lot of sense to think about how often do you sample. So a one sampling time point is usually not something you want to have. You want to have prospective samplings and so on. So there is a lot of unmet needs to standardize population and intervention trials. And the second level of I would say variation is tools, and there is limitations related to tools. And I come again back to this microbial diversity and how this is classified at different taxonomic levels, from the phylum to species and strain. And at the moment we use molecular tools based on molecules which are conserved, and this is the 16S ribosomal RNA gene, where variable regions are sequenced, or you sequence pieces of DNA. And at the end of the day, I think this is well established and give you a lot of insight to the composition and function. And here, if we have genomic DNA, we can even go to strong strain level analysis. Nevertheless, there is little side aspects. This is the 16S molecule, which you can see. Again, our analysis from the Cora cohort, and we compared, for example, different regions in the 16S molecule you can use to amplify, and different groups use different regions. For example, V1, V2 region, or V3, V4 region. And we compared the two, and what you see is, again, the richness level. If you use V1, V2, you see a different richness diversity between the two analysis systems. So having said that, none of the two is wrong, but the two basically give, if you put them together, they give a more complete picture. What you also see is if you use different platforms of analysis, again, this is basically a principal coordinate analysis, and the far away these dots are, the more different the ecosystems are. This is basically analyzed, standardized samples on different platforms, and what you see is that indeed the different platforms give very different results. And again, if you use different V regions to sequence your bacterial DNA, again, you have differences. Again, conclusion, sampling, sequencing, bioinformatic analysis matters. Conclusions sometimes are difficult to compare between different studies. The problem is not that this is basically wrong, what people publish when there is controversial results, but they may complement. So it's difficult at the moment to draw conclusions. And one of the recent, I would say, examples of confusion and controversies in the field is the question, is the placenta colonized, yes or no? And there was a couple of years back basically a publication which postulated that the placenta is colonized with bacteria. And this is very provocative, because me personally, I still believe in compartmentalization, and good fences make good neighbors. And the placenta is a very critical organ. And they postulated that within the placenta there is colonization, which may then lead to early colonization of the fetus after birth. This was revisited this year, and actually using a very detailed analysis. Take-home message is, no, the placenta is most likely not colonized. And this has something to do that placenta samples are very low in density of bacterial entities, and low dense bacterial ecosystems are very difficult to analyze at the level of molecular analysis, and there need to be a lot of controls to be involved to make the right conclusions. This was done here, and the conclusion was the placenta is sterile. So these are the controversies based on technologies. Most importantly, I think in the last couple of minutes, I think we need to talk about causality, because mostly what we see at the moment is associations. So we correlate statistically, for example, a disease with changes in the microbial ecosystem. This could be linked causally to a certain situation, but it could be also bystander changes. So the question around causality and function is very important. And for this purpose, in most cases, we need model systems, one of which is germ-free mice. So you can keep mice under germ-free conditions, and then you can colonize them and basically ask the question, if you have a model for a disease, if it's germ-free and still it's healthy, then the bacteria may play a role. Then you colonize the mice and you trigger the disease. One example given in the context of chronic inflammation is here. So we work here with a mouse which becomes a Crohn's disease-like inflammation. The inflammation is very similar to humans, so not everybody is getting inflammation, but a good proportion is getting inflammation, and this is basically the disease activity which you see here. That's under normal conditions in a specific pathogen-free environment. If you make the mice germ-free, then you basically completely abrogate the disease. Then, following Koch's postulates, and that's very important, you can take now examples from this condition, and you can basically transfer them back into germ-free susceptible mice. And what you can see here, the wild-type mouse is not getting the disease, supporting that this disease is triggered by genetic susceptibility. But in the genetically susceptible model, you can transfer nicely disease from the rat, which have disease. If you take stool samples from the blue mice, which did not develop disease, even though that this is that this is in a genetic susceptible environment, and you can see you cannot transfer the disease. So this follows Koch's postulates and clearly shows that bacteria drive the disease. You can do this in human settings. So this is a cohort of Crohn's disease patients with active disease and inactive disease. You cannot clearly separate this at the level of sequencing. When you now take samples and you transfer them basically back into the mice, you can see that you can transfer the disease with samples coming from active diseased patients. And if you take stool samples from inactive patients, the mice do not respond. So this is again a nice example how you can basically use models to show causality. In real life, there is one clear application. This is fecal transplantation, where stool samples are taken from certain donors and are transplanted back into recipient humans. The best example that this is clinically relevant are actually at the moment infections with Clostridium difficile. This is enormously effective in patients with refractory disease where normal medication is not very successful in treatment. And here, fecal transplantation is in over 90% of the patients successful to clear the disease from the patients. So there are examples where you can transfer basically a certain protective environment into a clinical setting. The question now is for which patients fecal transplants basically are useful. And then the second question is, what is it you want to transfer? Which is the best donor for a certain repertoire of recipient patients? So last two slides here, coming basically to the understanding of functions. And what you see here is basically a study where we used humanized mice, which I just presented, from Crohn's disease patients, and you measure a metabolic environment, and then you combine basically data. And the importance here is in this statistical analysis using machine learning, so we can classify disease based on bacteria only with a certain success. But if you combine networks between metabolites and bacteria, and if you combine this information, then you dramatically improve the classification, meaning that you can separate and model diseased from non-diseased people. So take-home message at the end of the day, many pieces of information need to come together from humans, a mechanistic understanding. You combine different information based on microbiome, based on metabolome, And you will hear a little later a bit more about the metabolome. You can combine this at the level of analysis to make basically machine learning and selection criteria and identify biomarkers. At the end of the day, you always have to go back and understand causality. So much has to go through model systems, and these gnotobiotic mice is one tool to basically validate biomarkers at this level. This is a long way to go, and my estimation is that we need for a couple of more years, so now my computer is frozen, that we need a couple of more years to understand all the different association data at the moment associated with published microbial changes, and the question, how much can you translate into clinical situations? Variation of patients and normal healthy individuals is large. We need to standardize methodologies, and then maybe we can apply this to a risk estimation for diagnostic profiling, for example, and that's already clear, for certain applications you can use this therapeutically. So, take-home message, every individual basically has a very individual fingerprint of microbial profiles. These fingerprints will impact on the host, probably also in very individual ways. And to put this into context of health is very important. We need a reflection to disease. The complexity is high, and this is why there are still substantial knowledge gaps in the interpretation at the moment of the published literature. So, having said that, many people are involved in those studies, and my guess would be that we need another 10 to 15 years to clearly separate associations from causality and to clearly define clinical applications where we can use this information for patients. Thank you very much.
It's your stage.
Thank you. Do you see my screen?
Yes.
Okay, very good. So I'll go on into another profiling area, metabolite profiles, and the area of food intake, how to find and validate biomarkers of food and beverage intake. However, of course, given the title of this webinar, I'll give it a twist towards the microbiome as well. Two different joint program initiative projects have been involved here, the Food Bowl, Food Biomarkers Alliance, and the Dynamic, one most on food biomarkers, the other one more on the microbiota. So there is a major problem in nutrition research, and that is we have not a complete idea of what people eat, and many people do not have that idea themselves either. So we have different instruments, most of them subjective, to figure out what are people actually eating. They can be food frequency questionnaires, they can be food diaries or 24-hour recalls or interviews, and they are all pretty time consuming, based on memory, and also later on you have to calculate intake of different nutrients on food composition tables. So the question is, could we get closer to know what people are actually eating? So could we avoid the systematic and random errors caused by this? And of course, biomarkers of intake, so measuring metabolites that are related to certain food intake in urine or blood or fecal sample might be providing some new tools for actually giving us the idea what exactly was this person eating. And so we set out in the Food Bowl Consortium to try and say, okay, what is biomarkers? So we published this paper on the flexible classification system that is very important in order to understand how do we define food intake biomarkers. And very clearly, they should reflect mainly what you've been eating and not so much the effects, because several different foods would have the same kind of effects. And also the host plays in. So what the host contributes is a separate class of biomarkers. So with this definition, which is very much related to how you use a biomarker and not so much the measurements as such, then we went to all the different food groups and said, okay, we would like to know all the biomarkers for these different food groups and go on from there to subgroups and specific foods. So a large number of reviews of currently known biomarkers for these foods were published. Then thinking of metabolomics to help us on finding these biomarkers, we try to design a way of finding as many biomarkers as possible and to try and identify them using metabolomics. So the food metabolome here is defined as the impact of food that we eat on the human metabolome. You have other metabolomes like endogenous metabolomes, drug metabolomes, but this is what we are interested in this connection. So this, of course, depends on the sample type and also on the condition of the person as you sample. So therefore, the timing since a meal, for instance, and your own metabolism, and of course also your microbiota can influence this. So one of the tools that we've been using extensively to try and dig into the areas where there was a lack of biomarkers, especially foods that are often consumed but where there are no very good biomarkers at the moment, was to use meal studies. And they can be very useful design for finding putative biomarkers of food intake. So typically you give a food to someone who has been asked not to eat it for like 24 hours or 48 hours, and then you sample at several time points afterwards and analyze in an untargeted fashion which metabolites are changing as a function of time after the meal. You also have, of course, a control group, so that it's not just the postprandial condition that is affecting you, but the food as such. So using this, you can do metabolomics and try to find all the markers changing. And sorry, I go back again. From this, with advanced mathematical analysis, multivariate analysis, you can find the markers and identify those that are your putative biomarkers of intake. However, this is not giving you a final answer. You still need a lot of validation in order to know what you're doing. And we set out a scheme with at least 8 different characteristics of validation that you have to go through. Sometimes you can do several of them in one study. Sometimes you have to do several different studies in order to reach this point. A way of showing it is this drawing that was made by one of my students, Natalia Vasquez, when she was working on banana biomarkers, and the minions here are a lot of checkpoints. So you have to do these meal studies as the first step. You do, for instance, urine collections, you analyze them, and you find a range of markers. You start validating, typically in a cross-sectional study, where people have very different diets, and there should hopefully be no other food that gives the same biomarkers. And if this is the case, that you see the marker other places, it's not necessarily very specific, but sometimes you can combine markers, taking 2 or 3 markers together, and you find a pattern that is really good, and you can validate it according to all of your parameters. So this is the goal, and setting this up, we have tried to do that for several different foods. So how do they perform? Here's maybe one of the worst examples, maybe also expected to be one of the worst. That is looking at reported alcohol intake and well-validated alcohol intake biomarker, ethyl glucuronide, in a study here, cross-sectional study of 139 subjects. So now we don't know whether they have other sources of alcohol or whether they just don't know how much they are drinking. But obviously there is a trend actually in both directions, but towards some people having a much higher exposure than they think. This is maybe also fitting what we would believe, but still it's an important finding that needs further exploration. If we combine and look at two different biomarkers done on the same samples in independent measurements, so this was done on two different instruments, two different samplings, then we see that they are very well in concordance. So you have better than 0.9. So it's not the marker; it's definitely the dietary instrument that I also started with. So going on from this, for banana, for instance, we had to have a multi-marker, and you can see we also reach around 0.9 in concordance between whether people say they eat banana and they actually do it. So for banana, there seems to be no abuse, and they are actually able to report, although it never becomes perfect, also because we have inter-individual variation in metabolism. You can also see that the biomarkers here, they are common metabolites in humans, but the combination of them makes them unique to banana. For meat, this is also an area that is really difficult. And here we have had to divide the problem into several steps, saying, okay, we have to divide between omnivores and vegetarians, then between red and white meat, and then after that we can start quantifying the amount. Doing this in a stepwise approach, we actually managed to have pretty good prediction models, and in this way we can ask and divide people into different groups. I know very well that, of course, people eat several kinds of meat if they are carnivores in a day, and there are still things to be resolved in this. Then to the gut microbiome, because, of course, a lot of these foods, they are metabolized by the gut microbiome, and that leads to a range of different compounds, some of them specific to certain foods, and therefore you can see, for instance, whole grain, you'll see enterolactones, you'll also see more of the short chain fatty acids, you can see fruits, you can see meat that have a range of metabolites that are being affected. And as these metabolites are formed, they also have effects. So now we go from not only exposure biomarkers into effect biomarkers, where they actually start affecting the receptors and having affecting signaling in the human body. One of the studies in the dynamic consortium was the UNINA intervention that took place in Italy, where a range of subjects were either randomized to a Mediterranean diet or control diet, and we did metabolomics of fecal, urine, and serum samples for all these subjects at several time points. Here I've labeled with red what you see in blood, with yellow what you see in urine, and later you see brown. You can guess what that is. So we have markers of proteolytic degradation. So we see some indications in both groups that they actually increase certain fragments of proteins, but in general we see a lower level of protein degradation products after the Mediterranean diet. So as you see here, for a range of different metabolites, also some like paratresol sulfate that may at the same time have some effects on the kidneys, for instance. There is a lot of crosstalk, specifically between tryptophan catabolites and the host. so we were particularly interested in that. Some of them are beneficial, some of them maybe have adverse effect and be remotoxin, and especially the beneficial ones may affect a better gut lining and be immunomodulating. So among these many compounds, we found several in the urine, but in general we saw a decrease with the Mediterranean diet, or no change, I'll say, from baseline and until later. Whereas for those who had the more unhealthy diet, we see an increase in some of these compounds. Interpreting this in terms of health is premature, and some of these metabolites are very likely to have both beneficial or an adverse effect, or be quite neutral. So, but it's just a view into a window that we will be able to interpret much better as we go on with the research in the field. One example more is that in this intervention, nuts were the major source of lagic acid. So lagic tannin degradation products called urolithins were seen and very much increased in the Mediterranean diet. And these compounds are also thought of as compounds that could affect improved health, although good final proof for that is still missing. Whole grain intake, which of course whole grain was one of the things that we expected to see being increased, and we have a range of biomarkers. Some of them have been annotated since this slide was made, but several of them are still quantitatively annotated. You can see both in urine and in plasma that we see metabolites that go up with the Mediterranean diet, both at 4 and 8 weeks, whereas they do not increase or are unchanged with the normal or unhealthy diet. So we see also some new markers that need to be validated in additional studies. So to try and conclude on this, I'll say that by accurately classifying and validating combining biomarkers, it's possible to classify the recent intake of several foods and drinks correctly, and the number of foods and drinks that you can classify is ever increasing, although most of them are not finally validated yet. We can identify misreporting in short-term dietary assessments, so we could see people who are not following the diet they were supposed to do, and we can also correct in the statistical analysis for whether people were actually following the diet or not, which is probably could be a major leap forward in nutritional science. Then we also identify a range of food intake related biomarkers that are affected by the microbiome after dietary change. In general, I would say they are more interesting in terms of effects than in terms of exposure, but the combination is of course also important because these markers will be very different between subjects depending on the functionality of their microbiome. So the support for this was from the Joint Program Initiative, as I mentioned, also EU grant, and the Carlsberg Foundation have been funding part of this research. My group has been very much involved in all the development of these biomarkers. I've mentioned some of the names here, and I specifically showed some data from Natalia, Catalina, and Henrik Royer, and Rasto Monasek here. So I'd like to thank them, also football collaborators, and the Dynamic Consortium who are involved here. So I'm ready to take any questions.
To Professor Martin van Bergen from the Helmholtz Center in Leipzig. Martin van Bergen is giving a talk on metabolomics for functional analysis of microbiome host interaction.
Hello everybody, welcome to this webinar. And as you can not see in the moment, I guess, because it was not on the right screen. Now it should work. You cannot see me, but this is what I look like. Thanks God not. But I guess we also have to be careful, and we always are responsible for how steep or how flat the curve of infections will be, because at least in Germany we simply do not have sufficient intensive care units if the curve becomes too steep. So I guess it was a good idea to have this here now on as a webinar organized. So if you want to see how I look like, this is just a picture from 2012, so I look pretty much like that.
We cannot see your screen, Professor von
Yeah,
Bergen. Can you share?
I don't. Wait a moment. Okay, wait a moment. You cannot see my screen. Okay, I try again. This because perhaps I have to select this correct screen. Now it should work. Yes, now
Yes.
do you have it?
Excellent.
Okay.
Good.
Okay.
Thank you.
Thanks. So just start again. The joke was that actually I do not look like that. So we have to be serious, and this is what I look like today, more or less. So as the color mentioned and described quite nicely, we have a microbiome hype, and this is just an example that in a paper from 2013, Smith and al. Described for how different diseases associations with the microbiome have been found. Nearly it can be found for all different types of diseases, and the list is actually much, much longer. And we have to be pretty cautious between and make a difference between the statistical correlation between a detected microbial taxonomy found here on the right hand side and the disease X. And with all the statistical tools, it's always a little bit confusing, and especially if you do not use correct, for example, multiple testing adjustments, then it's easy to find a statistical correlation between the taxonomic data and the disease. So, and it's something completely different between if you have a statistical correlation or you have a functional correlation or even a functional causality. So in order to prove that, and this I would, in that sense, I would support the arguments from Dirk Haller that we have to be careful for that, and that we have to have more systems in order to prove the causality for effects in the microbiome and the association to the disease. And the second point here I want to mention is that also model systems have advantages and disadvantages. Of course, if you transplant human microbes into a mouse, they face there different host factors which are not present in human gut but are present in the mouse gut and vice versa. So, but as long as we stay aware that every model system has advantages and disadvantages, I guess we can make progress. So another thing is, and this was also part of the Q&A session with Dirk Haller, the question: how can we predict something from taxonomy? And I'm not so sure if we can do it all, because there's quite a long way from the abundance of a bacteria to its actual activity in situ. And the same is true not only for 16S data, but the same is also true for metagenomic data. Yes, you have there a quite detailed descriptions which genes are there, but you have no idea what the bacteria really do when they are in the gut. And most of the predictions are based on experiments which were performed on bacteria in single culture. And we always have to face the fact that microbes in the gut are not alone. So it cannot be nicely predicted, at least in my opinion, what a single or what bacteria they do when they are together in a consortium. So in order, having said that, one has to think what kind of methods do we have to use to get the information we want. And just to get some more explanation for that, we have on one hand the taxonomy, which we can describe either by a metagenome or by 16S RNA, and then we get the distribution of the different species in a consortium. And on the other hand, we have the different functions which are performed by the bacteria, which can either be described by the metatranscriptome or a metaproteome or the metabolome. And the good thing for both metatranscriptome and metaproteome is that you obtain simultaneously information on the function which is actually carried out by a gene, which is then transcribed into a protein. And due to the sequence and the huge databases we have in the moment, you also get the information which bacterium produced either the transcript or the protein. This linkage between activity and taxonomy is given only for transcriptome and proteome, which is not possible for metabolites. But the metabolites, and therefore they are here the circle around the different bacteria. By them, you can describe the overall metabolic activity of the different species in a consortium. And the good thing, again, also on metabolites is that it's really close to the actual functions of bacteria which are there. And it can be a little bit more complicated, as we can see on the right-hand side. There even, now it was gone, so now it's here back again. So you have the different techniques I described already, or which were also described by the Tyler, the 16S RNA gene sequencing, metagenomic approaches, metaproteomics, metabolomics. But then on that metatranscriptome, but then you can also use, for example, if you want to have information on the metabolic activity and on metabolic flux, you can use the stable isotope probings, like, for example, DNA, nanosins of fish, or protein, which allows you to pinpoint really the activity of bacteria when you add an isotopically labeled substrate, which is then taken up, and you determine either the incorporation into the DNA, the RNA, or the proteins. And then you can say, this bacterium took up such and such percent of its carbon from this source, and thereby you can distinguish between active and non-active bacteria in a consortium. So, having said that, I now would like to focus your attention on the metabolites that are crucial for mediating the microbiome host interactions. So, if you take the gut system or the different bacteria in that, we have some well-known, also for a long time known, metabolites like the bile acids and also the short-chain fatty acids that are crucial for the metabolic interaction, especially the hepatocytes. These counts for the primary bile acids, which are produced there and then given up and then are modified by the bacteria in the gut and then flow back into the liver and then also again have some effects there. The same is true, or it's also true that the short-chain fatty acids, which are taken up by the by the epithelial cells, are then also important for signaling because they can have an impact on the differentiation of the different types of immune cells. So I guess we have two major factors or two major part and organs of which are affected, which then turn up into the host metabolism or to the immune system of the host. And these are the main factors. If you want to have a closer look on that, you know that you have the primary bile acids, the cholic acid and the folic acid, which are then modified by the bacteria by dehydroxylation on certain sites of the bile acids, and this has a strong impact on the hydrophobicity, for example, of the different bile acids. And this means that extremely hydrophobic bile acids can also disturb membranes, and this is both important for bacteria and also important for the host, because if the cells if they have a problem with their membrane integrity, they also this can lead to, on the host side, to inflammation, and on the side of the bacteria to the cell death. And this means, and also they have, besides the physical activity or biochemical activity, they also have, especially bile acids, have a strong impact on other organs. Here you can see from a paper Shapiro et al. In 2018, they described that bile acids do not only work on the hepatocytes, but they also, for example, bind to the TG5 receptors in immune cells and also in a set of other cells in the body. So they can have a simple metabolite like bile acid can have wide range systemic effects on the human body. And the second pretty much important receptor for bile acids is the farnesoid X receptor, which regulates bile acid, lipid and glucose metabolism, and is of really great importance for the overall metabolism of the host. So Biocratis, we detect the bile acid in our experiments with the kit from Biocratis, which is here shown as an example from that. It has a lot of pros because it's a ring-tried, validated method. It has internal standards. It covers a good range of human bile acids, and with the instruments available at the UFZ, it's nicely to measure them. The con is of course that it's expensive, and that some other assays cover some more bile acids. Nevertheless, we will use that kit, and also we will compare this in an effort to standardize the methods in the framework of the DFG SFB 1382, which is located in Aachen, with different partners which use different techniques in order, and we hope that we will come up with a standardization, and especially which will then also allow the comparison between data from different groups. Especially, this is just a small field, but at least for data on bile acid quantification. So another very important set of metabolites that mediate the microbiome host interactions are the short-chain fatty acids, which are produced by the bacteria under anoxic conditions, and which can be taken up from the host, and especially butyrate feeds the epithelial cells for about 60% at least. So they need them, and there are several of them, starting from shortest acetate over propionate and butyrate. You have heard about that also in the talk from Lars Dragschnitt. And these, there are different pathways which the bacteria can use to form short-chain fatty acids, and they are described here. The important thing is that it's hard to predict which pathway is used. For example, only on the basis of the metagenome. You should have metatranscriptome or metaproteome data in order to confirm that. And then, of course, the question is, what do they do? What do they do in terms of the metabolic system of the host and in terms of the immune system? So we have established a method for that. We're using the same machine as we use for the bile acid in this case. And it's nice that we have this assay. We also have an assay for the long chain fatty acid, because this here is only up to 10 carbon atoms. We have also assays available for long chain fatty acids up to C20. Disadvantage of the method is that it's sometimes hard to estimate beforehand which concentration we have to use in order to get meaningful data. So I would like to introduce you to one piece of research which we did in the framework of the DFG SFB 1052 here in Leipzig, which is dealing with the effect in the field of obesity. And one last resort which you can go to in order to efficiently treat extremely severe and morbid obesity is bariatric surgery. You have here on the left-hand side the normal structure of the gastrointestinal tract. And if you want to change that by bariatric surgery, especially Roux-en-Y gastric bypass surgery, you disconnect here and then reconnect this part together with that part, and you circumvent the stomach and the part of the duodenum, and so you shortcut this, and this leads then of course to reduced intake of food and malabsorption of nutrients, because you don't have the enzymes which work already here in the stomach and in the duodenum, and you don't have this part of the gut which then also is responsible for the uptake of some nutrients. So it's a very drastic treatment, and it's only used if the person is really morbidly severe obese, because it's hard to predict the long-term outcome of this treatment. This has not been estimated so far pretty well. So, but if you don't have any choice, then people go for also for drastic treatments. So what we have done, we used rats in this case, and they were adjusted to some chow diet, and then we applied a high-fat diet at the age of 9 weeks, and then they got surgery. They got the surgery and they got a normal chow diet after surgery. And finally, they were sacrificed 16 weeks after surgery. And in this case, in this time window, they were observed, and we used only those rats that did not change the weight. Because the problem is, if you compare the normal rats after surgery, they lose quite a lot of weight, and this of course alters the microbiome as well. So we, in order to extract only the impact of the altered structure of the gastrointestinal tract, we took only those rats which did not alter, which did not lose weight. And then we did a lot of experiments. We sampled different parts of the gastrointestinal tract. We did the 16S, 16S RNA sequencing. We did metaproteomics and metabolomics for some part. And the interesting thing was that it turned out that the bile acids were quite dramatically altered. When we measured in the colon, you can see here on the left-hand side of this volcano plot that all the secondary conjugated and all secondary and all bacteria, the modified bile acids were altered quite dramatically. And this was linked to a change in abundance of bacterial taxa. Also, some of them are known to metabolically alter bile acids, but it was especially observed that less bile acids lead to a decrease, lead to an increase in bacteria, for example, those here, actinobacteria, which are susceptible to bile acid toxicity. And this means that when you have less bile acids, those bacteria which are affected by the toxicity of bile acids, they have a chance to grow, and this is what we can see here. Another aspect that we found was that there was a strong impact on the arginine synthesis. So here we have the combined data of the different metabolites which we measured also with the BioCraters kit here on different parts of the arginine pathway, and we confirm that with metaproteomic data on the enzymes which are involved in some step of those. So these are here shown in red. So we can combine metabolomic data and metaproteomic data in order to prove our hypothesis or the evaluation of the data and say that really the arginine pathway is involved. We also try to do this with a larger correlation analysis, and this works for somehow, but not always so good. What is quite nice is that you can also distinguish in these networks between the module hubs and the network hubs. So this helps a little bit, but still this picture is much too complicated in order to draw simple conclusions from that. And I guess this is always what I say is our crime, or in Germany it's a Tatort movie where people are always seeking to find the bad and the good guy, especially the bad guy. And I guess in the future we will have to get used to more complex pictures and more complex interpretations. There will be not one single good and one single bad guy, but there will be networks of functions that are required to fulfill the function of a healthy microbiome. And with that, I would like to come to the conclusions. I guess that metabolomics is quite at the center for assessing the microbial activity. It's quite useful to integrate metabolome and metaproteome data, and it is also it should be done more, but I guess especially based on experimental data to perform correlation analysis between metabolome and metagenomic information, because this provides hints for microbial ecology. And with that, I would just want to show that we are actually working on several other metabolites which might be important for microbiome research, like sulfonates, together with Professor Blaut in Potsdam, TMAO, which has also been described by Lars Wachschild. And of course, what a major field is not to detect only the known metabolites, but also find new ones, which might be either biomarkers for the transformation of different nutrients, or might play a role in the metabolic interaction between microbes or between microbes and the host, and therefore we established some tools for untargeted metabolomics, which I would not like to go into detail today. And I would like to conclude with some perspectives which I think are important for microbiome research in the future. I guess we need the complex systems like animal systems, but we also need simplified models like these bioreactors we have now established here at EFZ for gaining detailed understanding. The second thing is, I guess, metatranscriptomics is in the moment something which is not often enough used, because it provides a much better coverage than metaproteomics. And with some tools, we use this specific tool there. It's not so difficult to handle. And I guess it provides a lot of information. And for determining really the actual activity of microbes, or especially in the microbiome host interaction, stable isotope probing for metaproteomics and metabolomics will provide insights into the metabolic flux which is actually occurring there, and I guess which is key for understanding microbial ecology. And with that, if you need a cooperation partner for functional microbiome research, just send me a mail. I would like to thank many people who were involved in especially in this gastric bypass study, especially Wiebke Penske here in Leipzig, and also Janice Panagiotou from Jena, Matthias Noll and Christian Bittler from Bayreuth, and here from the UFZ, also from the Department of Environmental Microbiology, Susan Miller and Thomas Hübschmann, and from my department, especially Sven Hange, Ulrike Krolle-Tamczyk, Christian Pedersen, Nico Dienlich, and Dirk Wissenbach. And with that, I would like to thank you and for your attention, and I'm happy to answer questions.
Okay, am I?
Yes, we can hear you.
Okay, perfect. So I think we heard 3 very nice talks amplifying different areas in this research, and I think it becomes very, very obvious that we are just at the start. One conclusion for me really is the level of complexity and diversity, and this holds true for the microbiome with bacteria, and we're not even talking about viruses and other microorganisms. You know, on the other hand, the metabolite profile. So on both hands, the bacterial profile and the metabolite profile, they are enormously complex. From bacteria, we just start to analyze strains, which are individual components which affect then the host. And on the other hand, we have, I think, a metabolite repertoire which is still completely, I mean, at the beginning to be unraveled. So I think we're facing a transition at the moment where we try via technologies to define, characterize the individual level of components which affect us. I think that's one theme which is ongoing at the moment, and it takes for a little while longer to actually finalize this analysis so that we can say, okay, what is the diversity at the strain level? What is the diversity of metabolite levels? And then the second, and that would be, I would say, the even more complex scenario, is to integrate this individual information into host functions. And here, I would not say that one or the other component affects the host more or less. I think bacteria in a wide range have important impacts on immune regulation, tissue functionality, and on the other hand, many metabolites affect the immune system. They translocate across barriers. You will find them in blood circulations and can detect bacterial metabolites in urine. So we will face these, I think, 2 hallmark questions at the end: defining complexity at different levels, and then to translate this complexity into a systematic analysis on how these different components would affect host functions. And the third level then is the interpretation. How much would that interfere with disease susceptibility, disease progression, to answer the question of whether we can use them as biomarkers? And there is actually not many biomarkers around on this globe. So it is and will be a challenging situation to define biomarkers which explicitly define risk and profile risk for a diagnostic scenario and then in a therapeutic fashion. So I think we're at the start, and it's hopeful, but we need more time to actually do our homework. I think that's my conclusion.
Okay, thank you very much. So thank you all for attending this webinar, and I think these are the perfect closing words from you, dear Kala, and we're pretty much looking forward to probably the next webinar or the next real-life meeting, the next symposium. And so with this, I would like to close the webinar and thank you for attending.
Thank you.